Hello, future biologists. My name is Jiwon Shin, and I'm here today to give you a 5-minute crash course lecture about two macromolecules, starch and triacylglyceride. First of all, I'm going to tell you about starch. Let's start off with a definition. What is starch? Well, starch is a complex carbohydrate. It's a long chain of sugar molecules connected by glycosidic linkages, which is basically the bonds formed via dehydration synthesis. In nature, y'all know that glucose, which is the energy of plants, is produced in the chloroplast by photosynthesis, right? Well, when extra glucose is produced and it's left over, they are stored in the amyloplast of plant cells in the form of starch. Then they will later be used as the energy source. So how does starch look like? This is the basic shape of the molecule. An interesting fact is that starch is made up of two types of glucose polymer chain, amylose and amylopectin. Well, amylose is a straight chain polymer of glucose that is connected with alpha-1,4-glycosidic linkages, which is just a bond that is formed between the first and fourth carbons in two alpha-glucose molecules. Just in case you don't know what alpha-glucose is, there are two different arrangements of glucose, and if the hydroxyl group OH is below carbon 1, it is said to be the alpha-glucose, and if it is above carbon 1, it is said to be the beta-glucose. Now let's get back to the content. One molecule of amylose contains over 250 glucose units. At this point, one interesting property is that since the alpha-1,4-glycosidic bond connects the glucose rings with an angle, it forms a helical shape. This allows the cell to store energy in a compact form. However, when it's in its crystallized form with hydrogen bonding making up the helical structure, it occupies the places where polarity might otherwise be able to occur in the molecule and allow it to solubilize in polar water. So basically, this is insoluble. Next up is the amylopectin. This form of starch is a branched chain polymer of glucose that is connected with alpha-1,4-glycosidic linkages and alpha-1,6-glycosidic linkages. So there are two glucose rings connected to one carbon-1 atom. As this macromolecule has a highly branched structure, there are more places for polarity to occur in the molecule and allow it to solubilize in polar water. As you can see, the two structures of the starch have some completely opposite properties. Because of it, depending on the ratio of amylose and amylopectin, the overall starch properties change. Generally, as amylose is 20% and amylopectin is 80% of the starch has both polymeric and branching nature, which allows the macromolecule to have relatively less solubility in water. Starch can be found in cereal grains, seeds of legumes, tubers, fruits, and vegetables. As stated earlier, the main function of starch is to store excess glucose for plants. However, as starch is a big molecule consisting high energy in the molecular bond and glucose molecules that we use in glycolysis and cell respiration in mitochondria, starch is an important energy source for animals, including humans. Now let's move on to triacylglyceride, also known as triglycerides. This is an energy-rich compound that serves as an energy storage in living organisms. As you can see from the picture, it's a lipid molecule with a basic structure that is consisted of glycerol and 3 fatty acids. Glycerol is the backbone of the triacylglyceride, which is a water-soluble alcohol, and fatty acids are chains of carbon atoms with a methyl group on one end and a carboxylic acid group at the other. These two chemical compounds have undergone esterification, meaning that the alcohol and the organic acid were combined by ester bonds and formed triacylglyceride and 3 water molecules. Furthermore, there are two types of triacylglyceride. If the 3 hydroxyl groups of glycerol are esterified with the same type of 3 fatty acids, the lipid is called simple triacylglycerol. On the other hand, if the esterification with different types of fatty acids happen, it is called mixed glyceride. Now I want to talk about some properties. The most interesting one is about its fatty acid. In plants, fatty acids are mostly unsaturated. As there is a bend due to the double bond, it does not allow the molecules to sit nicely against one another. Therefore, many vegetable triacylglycerides are in a liquid state at room temperature. On the other hand, in animals, the fatty acid components are mostly saturated. In these molecules, it has a straight structure due to the single bonds between carbons. This allows the molecules to comfortably arrange themselves right next to each other. Therefore, many animal triacylglycerides, like butter and meat, are in a solid state at room temperature. Furthermore, since the fatty acid components have long carbon chains, which are nearly nonpolar and low density than water, the triacylglycerides are nearly insoluble in water and also float on the surface of water. Now, you might wonder, why is triacylglyceride important after all? Well, triacylglycerides in animals and plant seeds function as an important energy source. For mammals like us, when we need them, they break down into a glycerol molecule and 3 fatty acid molecules. Then, the fatty acid combines with albumin, which is a protein in blood plasma, and is carried in the bloodstream to the sites of utilization. Other than that, for animals, triacylglycerides also serve as insulation and padding for organs. Well, that concludes this 5-minute crash course lecture, and I hope you all have a great Chuseok break, and goodbye.